Bellwater. I'm a senior lecturer in the School of Biological Sciences at the University of East Anglia. And this project that's going on at the Sainsbury Centre this evening, linked to transformation and sustainability, is the culmination of a project that I've been involved with colleagues from across the university over the past 12 months or so. And we've been aiming to develop projects that link across the void between arts and science. So really trying to engage some people who would normally be interested in art, to tell them a little bit more about science and engage them in science, and also make the scientists appreciate and understand the different media that artists work with, and to understand in a better way how artists get their message across.
Basically, we're here from the schools, the science schools at the UEA, and we're just trying to make the link between aspirin and willow, and obviously willow and basket. It's as far back as the ancient Greeks. They were taking willow bark to relieve pain, but it took, well, hundreds of years before anyone actually isolated the active compound. And once they had, we developed the drug that we've got today, which is used in all sorts of cases of inflammation and pain. But the willow itself, where it's isolated from, is also very good for helping us to clean up the mess from the chemical industry. So you can grow willow as a crop, and it grows so fast that you can pick up the toxins from the ground and then basically cut it down, take it away, and you clean up the ground. So willow baskets explains why we're here, really.
The particular focus of tonight's event, that from our point of view, so from the biology and science point of view, is to highlight how there are many areas of biology that link to what we see happens in the way that humans use baskets. We're very familiar with baskets and how baskets are useful to us, but when we look in biology, there are many areas where baskets and basket-like structures are used within cells or by different animals, different organisms, and those are some of the ideas that we've been developing in the exhibits that have been on show here tonight.
Perhaps one of the most accessible ways for people to relate structure to biology is with something like birds' nests. So we've got a range here of birds' nests, very different in their structure but very similar in their function, in allowing these species of birds to obviously reproduce and rear their chicks. So we can see something like this nest here from pendulum tits, which we don't find in the UK. But you can see on the left hand side this nest's been suspended, and the eggs will be laid and the chicks will be reared inside that nest. This barn swallow nest, I like to think that this trowel's just been abandoned in somebody's shed or barn, and the swallow's exploited the discarded tool and built its nest on the back of the handle, and you can see it's made out of what would look to us as clay, straw and feathers, but to a barn swallow it looks like a very comfortable and snug little nesting environment.
Trying to preserve what we can. Well, this is a classic English willow shocker made by David Drew, who's one of our best players, now lives in France. And this was from his exhibition.
These are little bacteria, but each of those little dots is a colony of bacteria. So it's got thousands and thousands of bacteria, all in a little spot. And if I put it back in here, what's different about these bacteria is that they've got a gene in them called GFP, which just stands for green fluorescent protein. And essentially what that is a basket-like structure. A protein basket that holds this little molecule inside it called a chromophore, and essentially what it does, and no one knows why it does it, or why the jellyfish that we originally found it in has it, but when you shine very strong blue light on GFP, the chromophore in the middle gets very excited and emits light, but it emits it in a different wavelength. So if we switch this on and shine a very strong blue light on it, the GFP, as its name suggests, actually glows really brightly green instead. And what's sort of initially started as sort of a really quirky but it's kind of obscure little protein has actually gone on to become something that's used in science, sort of basically across the board in molecular science. So because obviously just by looking you can tell that your organism's got GFP in it, then it allows you to do all sorts of cool nifty things where you can use GFP to study organisms. So we've all heard about genetic modification, where scientists have been sort of making GM foods that have certain genes in them that maybe make them grow bigger, or can grow in sort of more extreme environments. And the problem, of course, is that when you inject these genes into an organism, how do you know that it's worked? How do you know that it's there? And the easiest way to do that is if you put GFP in it as well, you can know just by looking, shine a blue light on it. If it glows green, then you know that you've put your genes into the organism and it's working, that it's actually producing the protein, the GFP protein, and therefore it's also going to be producing the proteins that you want. This is actually GFP. Well, it's not the real thing, but the little spirals around the outside are the actual basket structure. And that's just simply sheets of protein that all spiral around each other to form a little barrel. And in the centre is the chromophore, and that's held in position, perfectly in position, to do its business.
There are some very famous examples of fluorescent cats and fluorescent mice, fluorescent rabbits. And while some of these organisms have very good sound, technical, scientific reasons to develop them, for some of them the science has been a bit questionable, and there's been a number of difficult discussions as to why the scientists have got involved in developing these animals, and it's not always clear that there's been a sound scientific basis. But that's one of the reasons that we thought this was an interesting topic to highlight tonight, because it gets people talking about these somewhat difficult scientific issues. Is it right to develop a cat that's green, for example, when there's no obvious advantage to it being green? And by talking to different people, such as the people that come to the Sainsbury Centre this evening, we're able to get different points of view than what we would normally have from scientists who might be much more comfortable and much more familiar with this type of technology and applying it to these situations with developing different organisms.
If you put your gene, your GFP gene, next to the other gene that you're interested in, then you can see that when one particular protein is being expressed, your GFP will be being expressed at the same time. So you can track how and when and where proteins are expressed by just watching them glow under a microscope. And so this is tobacco plant, and we can tell that this tobacco plant has been completely transformed because every single part of its body is glowing green. So that means that basically every single cell in that organism is carrying the gene for GFP, and that's what's making it glow. And this is some beautiful artwork. You should definitely get a picture of this. That's just bacteria on a petri dish, and each bacteria has got a different type of fluorescent protein, so red fluorescent protein, green fluorescent protein, and all they've done is just sort of smeared the bacteria in an arc, and as they grow, they glow.
Basically, each of the panels is a speaker that I've built myself, and each one has its own dedicated mini amplifier, which is being powered by a 9-volt battery. And for this particular use of them, they're arranged in a nonagon with the 9th side open for people to go in and out. The source material that's coming out of each of the panels is numbers stations, which are kind of spy espionage broadcasts from shortwave radio that have been collected over a number of years, not by me. and the video element is 1950s propaganda videos from America. So it's all kind of got a vaguely Cold War feel to it. The number stations broadcasts happened, they began towards the end of the Second World War and kind of picked up towards the height of the Cold War and then have tailed off.
I am a beggar at your gates, scratching stones that the birds have thrown, picking seeds the sparrows drop. What will they have left? For it's left that is left of me, and nothing is right, and your house is empty and open to the cold. I will creep in at the window, touch the walls that you own, run my fingers through the dust, remember what I must not do, for it's sin that is inside me, but I'll let nothing out, and when it comes to claim me, I swear I'll go quietly.
So another aspect that was very interesting to explore in the exhibits that we put on here this evening was to try and get interesting images, or images that were visually striking, because we felt that this would be something that would be of interest to people who came to art galleries. They would be interested to see these striking images. But behind those striking images there is some important scientific theories, and we've tried not to dwell on the science too much in the exhibit tonight, but we are hoping that some of the people that will have seen the images and seen the exhibits will be interested enough that they'll want to find out a little bit more about the science, and not be scared off by the science. So that's something that we hope to achieve from our point of view this evening, just to engage with a few more people who wouldn't normally hear about the type of science that we've been discussing this evening.